Hi, we are going to talk about protein. Proteins are the most versatile macromolecules in living systems and severe crucial functions in essentially all biological processes. They function as catalysts, transport and store other molecules such as oxygen, provide mechanical support and immune protection, or generate movement, transmit nervous impulses, and control growth and differentiation. So we will focus on understanding what proteins do and how they perform these functions. Proteins are linear polymers built of monomer units called amino acids, which are linked end to end. The sequence of linked amino acids is called the primary structure. Remarkably, proteins spontaneously fold up into three-dimensional structures that are determined by the sequence of amino acids in the protein polymer. Proteins contain a wide range of functional groups. These functional groups include alcohols, carboxylic acids, carboxyamines, and a variety of basic groups. Most of these groups are chemically reactive. The proteins within these assembles can act synergistically to generate competitors that individual proteins may lack. Examples of these assembles include macromolecule machines that replicate DNA, transmit signals within cells, and enable muscle cells to contract. Rigid units can function as structural elements in the cytoskeleton or in connective tissue. In addition, conformational changes within proteins enable the regulate assembly of large protein complexes as well as the transmission of information within and between cells. Amino acids are the building blocks of proteins. An alpha amino acid consists of a central carbon atom, we call it the alpha carbon, linked to an amino group or carboxylic acid group, a hydrogen atom, and a distinctive R group. So the R group is often referred to as the side chain. With four different groups connected to the tetrahedral alpha carbon atom, alpha amino acids are chiral. So they may exist in one or the other two mirror image forms called L isomer and the D isomer. So only L amino acids are constituents of proteins. For almost all amino acids, the L isomer has absolute configuration. So there is evidence that L amino acids are slightly more soluble than a mixture of D and L amino acids, so which tend to form crystals. This small solubility difference could have been amplified over time so that the L isomer become dominant in solution. Amino acids in solution at neutral pH exist predominantly as dipolar ions, also we call it like deuterium ions. In the dipolar form, the amino group is protonated and the carboxy group is deprotonated. The ionization state of an amino acid varies with pH. In acid solution, the amino group is protonated and the carboxy group is not dissociated. And as the pH rises, the carboxylic acid is the first group to give up a proton, and inasmuch as its pKa is near 2. The dipolymer form persists until the pH approaches 9, when the protonated amino group loses a proton. We sort these amino acids into four groups: on the base of the general chemical characteristics of their R group. And the first one is hydrophobic amino acids. The simplest amino acid is glycine, which has a single hydrogen atom as its side chain, with 2 hydrogen atoms bonded to an alpha carbon atom. Glycine is unique in being achiral. So alanine, the next simplest amino acid, has a methyl group as its side chain. Larger hydrocarbon side chains are found in valine, leucine, and isoleucine. Methionine contains a largely aliphatic side chain that includes a thioester group. The side chain of isoleucine includes an additional chiral center. So the larger aliphatic side chains are especially hydrophobic. That is, they tend to cluster together rather than contact water. The 3-dimensional structures of water-soluble proteins are stabilized by its tendency of hydrophobic groups to come together, which is called the hydrophobic effect. 2 amino acids with relatively simple aromatic side chains are part of the fundamental repertoire. Phenylalanine, as its name indicates, contains a phenyl ring as attached to in the place of one of the hydrogen atoms of alanine. So tryptophan has an indole group joined to a methionine group. The indole group comprises 2 fused rings containing an NH group. So phenylalanine is purely hydrophobic, whereas tryptophan is less so because of its NH group. 6 amino acids are polar but uncharged. 3 amino acids, including like serine, threonine, and tyrosine, contain hydroxy groups attached to a hydrophobic side chain. Serine can be thought of as a version of alanine with a hydroxy group attached, and threonine resembles valine with a hydroxy group in place of one of valine's methyl groups. And tyrosine is a version of phenylalanine with the hydroxy group replacing a hydrogen atom on the aromatic ring. Threonine, like isoleucine, contains an additional asymmetric center. Again, only one isomer is presented in proteins. So in addition, the set includes asparagine and glutamine, 2 amino acids that contain a terminal carboxyamide. Cysteine is structurally similar to a serine, but contains a thiophyl hydroxy and a thiol, short for SH group, in place of the hydroxy group. We turn now to amino acids with complete positive charges that render them highly hydrophobic. So lysine and arginine have long side chains that terminate with groups that are positively charged at a natural pH. Lysine is kept by a primary amino group and arginine by a guanidinium group. Histidine contains imidazole group, an aromatic ring that also can be positively charged. With a pKa value near 6, the imidazole group can be uncharged and positively charged near natural pH, depending on its local environment. So here shows negatively charged amino acids. And this set of amino acids contains 2 with acidic side chains, like aspartic acid and glutamic acid. These amino acids are charged derivatively of asparagine and glutamine, with a carboxylic acid in place of a carboxyamide. Aspartic acid and glutamic acid are often called aspartate and glutamate, to emphasize that at psychological pH, their side chain usually lack a proton that is present in the acid form and hence are negatively charged. So 7 of the 20 amino acids have readily ionizable side chains. These 7 amino acids are able to donate or accept protons to facilitate reactions as well as to form ionic bonds. So the table shows here gives equilibrium and typical pKa values for ionization of the side chains of tyrosine, cysteine, arginine, lysine, histidine, and aspartic and glutamatic acids in proteins. Amino acids are often designated by either a 3-letter abbreviations or a 1-letter symbol. These abbreviations and symbols are an integral part of the vocabulary of biochemists. So first, as I said, they are diverse. Their structural and chemical properties span a wide range, endowing proteins with the versatility to assume many functional roles. And second, many of these protein amino acids were probably available from prebiotic reactions. That is, from reactions that took place before the origin of life. And finally, other possible amino acids may have simply been too reactive. For example, amino acids such as homoserine and homocysteine tend to form 5-membered cyclic forms that limit their use in proteins. The alternative amino acids that are found in proteins, serine and cysteine, do not readily cyclize because the rings in their cyclic forms are too small. Okay, that's all for today. Thank you for listening.